Hello, welcome back. Today we're going to be going over biomolecules and carbohydrates in particular. Hopefully you kind of have a background on carbohydrates. You should probably know that they're sugars. We use them for energy. There's simple sugars, there's complex sugars. But the main idea here is that you should probably know that they make up a main staple of your diet, and these are things that are very important for living things to exist. So carbohydrates are almost universally used an immediate energy source in living things. The energy that we get from these, we either use it right away or we store it for short periods of time, not long periods. But we also sometimes see carbohydrates used structurally, mostly in plants. Now, to understand carbohydrates, you have to know the monomers and the polymer, I guess. The monomer for most or all these carbohydrates are something called monosaccharides. A typical one is just glucose, but a monomer is called a monosaccharide. Two monomers together is called a disaccharide, and a longer chain of monomers together is a polysaccharide. Hopefully you're starting to see a little trend here. Mono means one, saccharide means sugar. Monosaccharide means one sugar. Disaccharide means two sugars because di means two. Poly means many. Saccharides mean sugar again. Many sugars. So monosaccharides are just ready energy. They're things that your cell will use for cellular respiration in all living things. So it's a single sugar molecule. So on the right, this is glucose, C6H12O6. It's a common monosaccharide. It's just a hexose, hex meaning six. Os is a Latin root for sugar, so six sugar, so six carbons. It's used most commonly in cellular respiration almost by all organisms. There's a couple that use a couple different kinds, but mostly it's just always glucose. Some other famous monosaccharides is ribose and deoxyribose. They're both pentose sugars, which means they have 5 carbons. Ribose is used in RNA and deoxyribose is used in DNA. So disaccharides are two monomers together. So in the case of maltose, it's actually two of those glucoses together. And we use, as humans, maltose as brewing. That's where it's famous. There's just two glucoses joined together by the dehydration synthesis we talked about. And you can reverse a dehydration synthesis by hydrolysis. These things are reversible. You can go back and forth. Sucrose is used in sweeteners. It's something that you guys might have seen on the back of your nutrition labels on your food or your drinks. And sucrose is also used in plant transportation of sugars. A lot of plants will turn the glucoses into sucrose. That way it's a little bit easier to move. And you it's like a mass movement type thing. Instead of just moving one glucose, you can move two at a time. Lactose is probably one of the more famous ones and something that you guys know about. You might know a lactose intolerant person. Now, what lactose is just glucose combined with galactose, which is just another kind of sugar. So individuals who are lactose intolerant can't break apart these disaccharides. They can't break them apart because they're missing one of the enzymes, which is lactase. ACE, A-S-E, is a Latin root for proteins, or we usually use it for enzymes. So anytime you hear something like ACE, it means they either break something apart or help a reaction get going. So lactose intolerant people don't have lactase, meaning they can't break down lactose. Now when you can't break something down, you don't break it down. It kind of sits around and it kind of becomes almost like a poison. So your body has to get rid of it. There's only two ways you can get rid of something. It's either in your stomach, it's either going to go up or down, and depending on the person, they can let you know how that happens. So those are disaccharides. Polysaccharides are energy storage molecules most of the time. There's two different kinds. There's starch and glycogen, but these are short-term storage. They're not long-term. We're not keeping these for a couple months or a couple years. It might be a couple just hours. And they're hard to get in the cells. We don't really move glycogen around in our blood. It's big. It would get clogged up. It just would be bad news. But starch is glucose storage in plants. They store it in their roots for the winter months. It will actually go bad if it's left in there for more than like 3 or 4 months. but starch is a little bit more stable than glycogen is. Glycogen is glucose storage in animals. After we eat food, insulin is released by our pancreas. It promotes the conversion of glucose to glycogen, because most of the time when you eat something, you're not really using it for energy right then. You're not like, oh, I need to eat this power bar, that way I can take a step. You're saving the energy so you can use it later on when you're doing other activities. So we translate a lot of that glucose into glycogen to store it for the day's use. That's what insulin promotes. It happens in your liver. It turns all these like little glucoses over here and combines them all into this weird and crazy chain that actually has a pattern, but we don't have to learn about it in this class. There's also another hormone that's released that makes that glycogen break back down into glucose. That way we can send it to cells through our blood so they can do cellular respiration. If you don't use your glycogen, it eventually gets moved somewhere else and gets built into fats. So polysaccharides can also be used structurally, like I said. And even though I said most of the time, almost universally, it's used for energy, the structural sense of the polysaccharide, cellulose, especially the most common one, is actually the most abundant organic molecule on Earth. It's something that you look out the window, you probably see all around you. Cellulose is a main component of wood. It's also a main component of a lot of leafy plants in their cell walls. So, I mean, it's something that's always basically around you. It's in plant life. Now, what's interesting about cellulose is animals can't actually break it down. And a lot of animals that you think can break it down can't break down plant matter. So like something like a rabbit or a cow, they can't break down cellulose by themselves. They have to have other microorganisms inside of them, and they also go through multiple stomachs. Like a rabbit breaks it down a little bit, gets rid of it through its bowels and intestines, basically poops it out and eats it again. That way it can break down the cellulose completely. Something like a termite is an animal, and we think about it as eating wood and destroying wood, but it actually can't break down cellulose. The little microorganisms in the termite are the things that actually break down the cellulose once it gets in the termite's stomach. Why the protists actually do that inside the stomach is because they have a safe little home. There's no predators in there. The termites like them in there because, well, they get the food from it. It's kind of a mutual relationship. Now, when you get to college, sometimes you guys can actually but you get termites, you bust open their guts and you look at the protists inside their stomach. Now, on the right, there's a picture of cellulose. This is just the add-on. I just find it cool. Is the structure of it is basically in sheets, but it's connected, and it's kind of just sheets laying on top of each other, back and forth, and it's actually one of the strongest structures known to man. We actually mimicked cellulose's structure to make bulletproof vests. So cellulose is a very, very, very strong structure. So that's what I'd like you to know about carbohydrates. Is these two things. If you know these two things, you got this lecture down pretty well. Remember, there's going to be a test in a couple weeks, or a week or so. These are things that are covered on the test. Make sure you know those things, and I will see you in class. Thank you for your time.